Good morning, my dear students. I welcome you all in this video tutorial regarding SOS repair of DNA damage, or this is called also error prone repair. We have already discussed in the class regarding types of DNA damage, the causes of DNA damage, how DNA is repaired, mismatch repaired, base excision repaired, nucleotide excision repaired, and there are several repair system. But in case of SOS repair, this is called actually SOS repair mechanism. Why mechanism? Mechanism because it needs a series of pathway. Pathway means there are lots of protein involved in this repair pathway, by which several genes are activated and get repaired the whole DNA sequence. That's why this is called the repair mechanism. So here are some few features about SOS repair. The SOS response was discovered and named by Miroslav Radman in 1975. This is important for MCQ also. In this year, this scientist was first identified this pathway. Okay, so first of all, I need to tell you that there are several proteins and genes are involved in this system. I will come one by one. First of all, the SOS response is a global response of DNA damage in which the cell cycle is arrested and DNA repair, mutagenesis are induced. During cell cycle, you can remember, I have already taught in the class, during S phase or synthesis phase, that means where DNA replication occur, at this stage, if DNA is damaged, that DNA needs to be repaired. Otherwise, it will be mutated or mutagenesis. So that means replication will not be there. So it is very important to repair the damaged DNA during cell cycle. The system involved the REC-A protein, that is also called RAD51 in eukaryotes. RAD in the name of the scientist name. The REC-A protein stimulated by a single-stranded DNA is involved in the inactivation of LEX-A repressor, thereby inducing the response. So these are the things stepwise you have to remember the protein name and the repressor name. Okay. REC-A protein, REC-A protein will stimulate the single-stranded DNA, and that is involved in the inactivation of LEX-A repressor. Okay. So another feature is this is a error-prone repair system because that is attributed to mutagenesis. That's why this is called error-prone repair mechanism. So now the steps is very important. The SOS DNA repair mechanism. The damaged DNA cause REC-A protein to trigger the response and result in the autocleavage of the protein called LEX-A protein. I'll show you in the picture detail. Here is the picture. I will show you later. Just you note down the steps. Then REC-A protein is activated on binding on a single-stranded DNA. Then LEX-A repressor. This is a repressor that participate in DNA repair. Okay. So REC-A forms a filament around this single-stranded DNA region in an ATP dependent fashion and becomes activated. So what it does, REC-A forms a filament around the single stranded region in an that is nothing but the ATP dependent fashion and becomes activated and followed by the activation of LEX-A protein. The activated form of REC-A interacts with the LEX-A repressor to facilitate the Lex A represses self cleavage from the operator. Okay. So now, next, once the pool of legs A decreases, pool means the sum of legs A complexes, the repressor, the number is decreases. Repression of the SOS genes goes down according to the level of, let's say, affinity for the SOS boxes. This is very important thing. SOS genes are very important because you can see, I have already told you, SOS DNA is a DNA repair mechanism because there are several proteins that involve to repair in certain way, followed by activation of genes, the SOS genes. So lex A decreases, then repression of the SOS genes goes down according to the level of, let's say, affinity for the SOS boxes. What are the SOS boxes? I will come later. In this slide you can see, then operator that bind, let's say, weakly are the first to be fully expressed. Okay. In this way, Lexa can sequentially activate a different mechanism of repair. Genes having weak SOS box, such as these are three genes are very important. UVR-A, UVR-B, and UVR-D are fully induced in response to even weak SOS inducing treatment. Okay. So thus the first SOS repair mechanism here, the term is mechanism is mentioned, to be induced in nucleotide excision repair, whose aim is to fix DNA damage without commitment to a full-fledged SOS response. So this is the overall step of SOS repair mechanism. Okay. So step is the rec A activation, then rec A will lex A will activate, rec A will cleave at the repressor position, and then SOS genes will be activated, followed by nucleotide based reparation initiation. This is the overall step. So in this picture also you can see the SOS response. Replication shall be DNA damage, replication stall and DNA damage. Rec A protein is activated, lex A repressor, and lex A auto cleavage. Then the SOS genes in the SOS box, UVR A, UVR B, and UVR-D, these three, they will be get activated and followed by initiation or inducing of nucleotide excision repair. Then DNA damage will be repaired. That is all about the SOS repair mechanism of DNA, damaged DNA. Thank you.